Homemade Food Preservatives by Escuela, Politécnica, Ingeniero, Jorge, Matute, Remus, and our team, Renewable Team. Our team is composed by
Sara, Cervantes, Espinosa, team leader.
Alondra, Quetzalli, Rodríguez, Candelas.
Our advisor, Yadira, Ivonne, López, Aviña, and me, Bustinduy, Contreras.
The program talks about the different systems that help preserve food. The homemade preservative has to preserve the greatest variety of nutrients like carbohydrates or protein. The considerations that you should take into account for better application of the product are that you have to apply a generous amount of mixture directly on the food. Next, you have to cover the food inside in a closed glass container with a stainless steel lid. And next you have to place in dark place with limited light. The general objective that we have is prolong the lifetime of variety of food, maintaining the quality including texture, flavor, and nutrient. And our specific objectives are analyze the chemical components contained in peppers and design an experiment to determine the preservative period of the homemade food preservative. Pepper is made up of several components such as carbohydrates, proteins, fats, fiber, among others, water stand up. It also contains vitamins B2, B6, and C, and some minerals such as calcium. The main component of interest is the capsaicin, that gives these peppers the spicy flavor.
Vitamin C, or ascorbic acid, is an important water-soluble antioxidant agent. It possesses acid properties and strong reduction activity. The origin of those properties lies on its enedible structure and the possibility of ionizing hydroxyl in the third carbon, resulting in an anion stabilized by resonance. It also regenerates and maintains the active state of vitamin E, which contributes to the antioxidant effect. The ascorbic acid content in red pepper is 132.6 milligrams per 100 grams minus 1 of formula weight. There is an increase about 50% in ascorbic acid content in red peppers during its storage, which can be attributed to the metabolism of carbohydrates and accumulation of sugar in mature fruits. Capsaicinoids are a mixture of monosubstitute primary amides present in most varieties of chilies. They are responsible for the spiciness. Capsaicinoids content in red peppers is a density of 1,525.68 of capsaicin and 2,372.5 of hydrocapsaicin. Capsaicinoids possess antibacterial activity against pathogens in addition to its antioxidant activity. Pepper extracts show greater power antioxidant in the day 25 because of the presence of the capsaicins and hydrocapsaicin. In addition, antioxidant activity of red pepper has been attributed to high levels of carotenoids, catechins, and ethers. It also to the synergistic action between the compounds and the system of conjugated double bonds capable of capturing free radicals. The total of carotenoids is 2,243.1 milligrams per gram of formula weight. Carotenoids have a quick shutdown effect in the transition of one molecule of dioxygen and three of them. They also prevent the energy transfer from excited state chlorophyll to three oxygen molecules. Therefore, they are suitable for the protecting fine oil containing fruit from type 2 photooxidation.
Now let's talk about the acetic acid. This acid can be found on the vinegar, most specifically in the white vinegar. According to UPAC, the name is ethanoic acid. This because these have just 2 carbons. The concentration of acid in the acetic acid solutions that can have antimicrobial effectiveness are around 5%. This can eliminate the microorganisms such as the Staphylococcus and the E. coli. In this experimental study, they applied apple cider vinegar on a F. fecalis bacteria. So just to have a conclusion, they prove that the antimicrobial properties of the acetic acid is due because it's very acid and acidify the medium where the bacteria grow. So this stop the reproducing and growing of the bacteria and eliminate the microorganisms.
Our team proposes to extend the life of preservative properties found in the structure of the red pepper by applying a heat treatment. In this study, the changes of vitamin C concentration treated by 706 watts in the microwave for 2 minutes keep the ascorbic acid concentration consistent. This because of the essence of a leaching process in the microwave treatment retaining a higher concentration of antioxidants. By applying a heat treatment, we get a water activity of 0.72 to 0.8. Foods with these water activity values are known as intermediate moisture foods that are largely protected against microbial spoilage. Based on the results of psychochemical properties in different pepper stages, we made a calculation based on the percentage of change between the green pepper and the pickled green pepper. Then we applied a proportion to the results of dry red pepper, giving us the results of pickled dry red pepper. We can observe the stability of the results and also the increase of acid properties that we are looking for. The team proposals led to the elaboration of a simple homemade mixture considered as GRAS, generally recognized as safe. The mixture preserved lipids, carbohydrates, proteins. Pretreatment, gritting, and the drying treatment, microwave, gave us a shelf life of 2 to 4 months. The pickle with acetic acid vinegar gave us a shelf life of 6 to 12 months. The team concludes the mixture gives a shelf life of 6 to 12 months without losing its properties. The texture of the food that is preserved can be more moisturized because of the vinegar, and the flavor can be acid because of the vinegar and the pepper, and the pungency is given by the pepper. The nutrients preserved are vitamin A, E, C, D, K, B2, and B6. The changes are in the vitamin B12 and the folic acid. Also, the color changed to a red pigment.
Finally, team members and me would like to give a special thanks to teacher Mario Vega, Adriana Delgado, and to our classmate Alondra Jasmine Carmona Hernandez for the checking of our project proposal and for the methods of teaching and the development of this study, but also this endeavor would not have been possible without the team too. Thanks.
Finally, we have the references used in the elaboration of the presentation. Thank you for your time.